It is a distinct pleasure for me to introduce this afternoon's speaker, the main event, so to speak, Dr. Hal Weintraub, who was born and raised in New Jersey and received his Bachelor of Arts degree from Harvard College in 1967 with a major in biology. He then proceeded to the University of Pennsylvania School of Medicine, where he received a PhD in 1971. From there, he went and was a Helen Hay Whitney Fellow at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England, where he worked under the mentorship of Sidney Brenner and Francis Crick, two of the seminal thinkers in molecular biology. Most of you will know who Francis Crick is from Watson and Crick fame. From there, he went to a position at Princeton University on the faculty from 1973 to 1977. In 1977, he moved to Seattle, Washington, where he is currently an investigator of the Howard Hughes Medical Institute at the Fred Hutchinson Cancer Research Center. He's an assistant editor of Science and Cell, two of the most important journals in the field. He is also a member of the very prestigious National Academy of Sciences. Some of his major contributions to the field have been the development and improvement of the technique of antisense RNA, which is a potentially very powerful tool for regulating genes in a directed manner. One of his other major contributions has been the discovery and analysis of the master regulator gene in muscle development, MyoD. So without further delay, why don't I let Dr. Weintraub take over, and the title of his talk today is From Egg to Organism.
I guess I should start by saying that I'm not Bob Weinberg. He'll be here in November. I've been at many meetings where I've been introduced as Bob Weinberg. It's very... what I'd like to talk about is how from an egg, a fertilized egg, you build a whole organism. The exciting thing is that to a first approximation, I think in the past, well, I started in this field about 25 years ago and thinking that this problem would never be solved. And amazingly, in the past 5 years, we know the basic outline of how you do that. How from a fertilized egg you build an entire organism. We don't know a lot of the details, but a lot of the players in the game, the genes that are involved and how they work, are actually now known. Now, the problem is illustrated on the first slide. I think I can control this. Can you reverse? So before I begin, it's really important that you interrupt me anytime you're not clear about something, because chances are someone else has the same kind of question. Most important, I'll make a lot of assertions because I can't go into all of the experimental detail. And it's important to always ask how we know what I say we know. So if that bothers you, just raise your hand and say, how do you know that? Okay, so the problem is this. All cells have the same DNA. So you start with a fertilized egg. It has 100,000 genes in it. It divides and divides and divides. One cell becomes a muscle cell. One cell becomes a brain cell. Yet those two cells have the same DNA content. Now, if the DNA is giving the instructions, as you all know, the question becomes, how can those two different cell types have the same DNA if they're different? So that's the ultimate paradox. Now, the answer to the question is illustrated here. The answer is that the cells are different because even though they have the same DNA, all of the genes are not working equivalently in both cell types. So for example, if you start here with a fertilized egg and it's using or expressing genes A, B, C, and D, it will divide, give rise to two different cell types. This may go on and become a brain cell, this a muscle cell. This cell will continue to make A, B, C, and D, and maybe this one as well. But this will turn on other genes, G and H, this E and F. So even though the two cell types have the same DNA, the DNA is not used the same. So then the question becomes how, during this developmental process, you turn on genes E and F in this cell and genes G and H in that cell. So that's the question. So then you should ask, well, am I really sure, are we really sure that the DNA is the same in both of those cells? That was the assertion. How do we really know that? Well, this is a classical experiment that was done, unfortunately not in my lab, where you take a fertilized egg, happens to be a frog egg, and they're so big that you can just flick out the nucleus of the egg. Of course, the nucleus contains the genes and the DNA. Flick it out, and then take the nucleus, let's say from a brain cell, and put it back into this guy so that it has a brain nucleus. And now ask what happens to that fertilized egg. And what happens is it develops into a whole frog. You can do this from many different cells in the body, and this, I think, is as good a proof as any that the DNA is the same in every cell and that it's used differently. And that's the basis for the differentiation between a brain cell and a muscle cell. Now, the implication of what I just told you is that usually genes don't work. They're there, but they don't work. And you have to do something to turn them on to make them work. By work, many of you know, means that the gene is copied into an RNA molecule, which is a copy of the gene. And when it's copied into an RNA molecule, then it's working. So we want to know why in one cell type we make this RNA, and in another cell type we make that RNA. Now, I'll go through a little bit of bookkeeping. Roughly speaking, there are about 20,000 genes per cell, 20 to 100,000. Of the 20,000, 15,000 are active in all cell types. They're what we call household genes. They make a cell a cell. They take care of ATP, cell division, and so on. About 5,000 would be cell type specific. They are the unique genes which make a muscle cell, and a brain cell, etc. And this is a diagram of what that looks like. That is, here's the blood cells. Can you see the pointer? Is that okay? Here's the blood cell genes, here's the brain genes, and here's the muscle genes. These genes are all made in common. These are the household genes. These are unique to brain. These are unique to blood. So that's about what it looks like. And that's what we want to understand, how that comes about. Now, there are about 100 different cell types in a human body, approximately. 50,000 genes per organ, and each cell expresses about 20,000 genes. Just a review again for those of you who weren't here earlier. Of course, DNA is a long polymer which can be depicted as such. In one cell, there's about actually an inch of DNA. So that much DNA is compressed into 1 micron in the cell nucleus. The DNA has 10 to the 9th positions in this chain. That's 1 billion positions. And of course, it's filled with the alphabet A, T, C, and G. What we mean by chromosomes is just the DNA isn't continuous, but it's split up into units. And those units are called chromosomes. On each chromosome, there are about 1,000 genes per chromosome. And since there are about 50 chromosomes, that makes about 50,000 genes in a human being. So of one of these genes, on the chromosome, one gene of the 1,000 genes per chromosome will have, when you go through the arithmetic, about 1,000 positions per gene in this polymer, A, T, C, G. Now, the uniqueness of one gene from another, of course, is in the sequence of A, T, C, and G. Any questions on that? Now, when we say a gene is active, what we mean is that this gene makes a copy of itself into RNA, and that gives a protein product which determines a function. And what we're really looking at is the function, that is the effect of the protein, but that's a representation of what gene actually worked to make the protein. Now, this is an example of the intricacy of this problem of how you make a given copy of a gene. You remember the genes are usually off, and you have to do something to them to get them to work. Here you see a typical gene. Within the sequence, there's a unique sequence which says start, and a unique sequence that says stop. What it means is start making RNA, stop making RNA. A protein sits on the start sequence. The protein is called RNA polymerase. It actually sits on the DNA physically. You can look at it and see it there. Okay? And it makes a copy of this sequence. And here you see it coming out of the protein TACGT, corresponding to that. Now this gene is active because it has made an RNA product. The big question now that we want to understand is, how do you direct this very neutral protein, RNA polymerase, to this gene to get it to work? And the answer is that you use a protein to bind nearby, again to a specific DNA sequence. The protein touches the DNA and physically reads the sequence, A, T, C, and G, and distinguishes it from A, C, T, G, because the chemistry is different. When this protein, which we call a regulator, binds, it stands there as a flag saying, RNA polymerase, you sort of dumb thing, come along here and make the gene copy. And the polymerase just follows orders and makes the copy of the gene. Now, I think I'm pressing this too hard. You then ask the question, well, where does RNA polymerase come from, and where does this regulator come from? Well, the only answer is it comes from a gene itself. That's the only place it can come from. So it is made also by an RNA, which is encoded in a gene, and it is a protein which happens to bind to DNA. So here you have, in this case, the polymerase making that gene. The RNA is made, it makes a protein, and the protein can go off into the cell and do lots of different things, but importantly, it can come back, bind upstream of its own gene, and say, keep on transcribing me, keep on making copies of me. This is a switch, because as soon as you make a little bit of this protein, it's going to come back, bind here, and keep this gene permanently on. So it's a positive feedback switch. And this is extremely important in biology. And I will argue and show you that the reason why a muscle cell always remembers it's a muscle cell is because it makes a protein, which we call MyoD. And that protein not only does a lot of other things, but among the things it does, it will come back and bind to its own gene and keep it on. And that's why suddenly in your body, your muscle doesn't turn, unfortunately for some, into a brain. So maybe it's happened the other way around. So this is the paradigm. The DNA makes RNA, the RNA makes a protein, the protein feeds back. And what it does is the protein feeds. Can you see the light? The red dot is working? That works better? That works better? So the DNA, so the point is the protein feeds back, and it can either bind to the DNA and activate it, or in some cases bind to the DNA and inactivate it. Now, this is a picture of a gene working, because you didn't believe what I just told you. It sounded like very mysterious. Well, here's the DNA. This is an electron micrograph. Here's the long piece of DNA. It's got a unique sequence on it. And you can't see the polymerase, RNA polymerase protein very well. It's right here along the DNA. It bound over there first. It's making a little bit of RNA copy, which you see as a little tail. And as you go along here, the tail gets bigger and bigger and bigger. And that's the gene being copied into RNA. This is a cartoon of that, where you say, start, the polymerase binds, it makes a little bit of RNA. Now this RNA corresponds, if you think about it, to this sequence here. Now it goes further along, and the tail gets bigger, because the sequence distance is much bigger, and so on. And this is two genes in a row. This is very hard for me to use. This is two genes in a row, and you can see that this is a so-called Christmas tree where the RNA stuck to the DNA happens to flip down on the microscope grid this way instead of all to one side. It's just flipping this way, and it looks like a Christmas tree. Okay, now how do we know? You should have asked. How do we know there are 50,000 genes? Well, the way we know is that you can count them. And there are many ways of counting them, and more or less the numbers come to between 50 and 100,000. Now, one of the best ways is that if you have an RNA molecule, which is a single strand like this, a single strand copy of the duplex DNA, and it has this sequence on it, what one can do is make on a machine, a DNA synthesizing machine, which you buy from Applied Biosystems, a piece of DNA. You can get a gram of it. And yeah. Absolutely, yes. So for RNA, the difference is that it uses something that looks like T, but it actually is called U, but it looks like T. And for simplicity, it's often easier to not make that distinction. Thank you. So here's the RNA. So we make a copy of the RNA with DNA, and as you all know, T pairs with A, T with A with T, G with C, and so on. Now, this process is called hybridization because you're forcing DNA and RNA to come together by these rules. And if they come together, we score it as a positive. That is, this gene is there and it's being expressed. Now, if we want to get really fancy, we can actually see the gene in the cell. The way to do that is to add a dye to this DNA, and that's done chemically. You take a little bit of blue dye or whatever, chemically add it to this chemical, this DNA chemical with that sequence, and then put it on a cell. And what you see is that the whole, if the cell is making that RNA, it turns blue. So we can count things that way. Now, the other way of testing whether or not a gene is working or not is with an antibody. Now, you all know that you get a virus infection and your body responds to the viral proteins by producing an antibody. This to study genes. They take a gene, inject it into a rabbit. They take the protein product of the gene, inject it into the rabbit. The rabbit makes it an antibody. So the protein would look like this. The antibody would look like this on the left. The two would combine with wonderful specificity, and now you just detect it. And you can detect that by putting a dye on the antibody. And if the cell is expressing this red protein, the dye would bind to it, and you'd light up the nucleus, if it's a nuclear protein. Okay? So we detect genes by either hybridization to the RNA or with antibodies to detect the protein product of the gene. Now, genes, because they produce protein products, which can be regulatory proteins which bind to genes, they can talk to each other. So one gene can turn off another gene or turn on a third gene. Here, for example, you can see this gene is active. That's this squiggly line. It's making an RNA product. The RNA makes this protein over here, which is yellow. This yellow protein can bind to the control region of this gene and keep it on. That's what I talked about before. That's a switch. This yellow protein could bind somewhere else by another gene, turn that gene on. Or it could bind here and turn this gene on. Or it can bind, let's say, in the middle of this gene and turn this gene off because it acts as a blockade. As the RNA polymerase is trying to go through the gene, it's going and transcribing, hits this protein, and it can't go any further. It's all very physical. It's all there. And there's nothing magical about it. You know, you put something in the road and it can't go any further. So, first point is that genes can talk to each other, turn this on, this off, and so on. Genes can also have multiple inputs. So, for example, here's a gene, here's the RNA being made. This protein, this protein, this protein, or this protein can all bind upstream of this gene, and all can tell the RNA polymerase to come in and copy me, make me active, make me produce things. So there can be multiple inputs. So for example, here, you might imagine a small hormone, progesterone. It will bind to a protein, pink, and pink will now bind to the DNA and activate the gene. ATP might bind to yellow protein. That would then bind to the DNA and turn off the gene. Similarly, orange with oxygen and so on. So a given gene does not obey the signals from just one input. So you can make all sorts of interesting conditional events. That is, this gene should not be on until both progesterone and estrogen are present. When estrogen and progesterone are on, you get 2 pluses, that means go. However, if ATP is around, then don't go. And this is the way genes actually work. They have a logic to them. They don't go on or off willy-nilly. They go on and off in response to specific instructions, which can often be yes, no, 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 yes. How the gene on the piece of DNA integrates someone telling it yes, no, no, yes is really a major question in the mechanics, the actual physical mechanics of the transcription process. Yeah. Yes.
Why don't you just get a piece of whatever it was trying to do and just make those pieces over and over?
Yeah. The question is, if you have a protein sitting in the middle of a gene and the polymerase is coming along making a copy, why don't you get small pieces of RNA? Well, you do, actually, but then the gene gets constipated because it's blocked over here. And what you do is you keep on putting more RNA polymerases on until you fill up the whole gene. And that's maybe 4 or 5 RNA polymerases, and then it just can't go any further. It can either just fall off or just stay there. Yeah, and stay there for hours. The point is that because genes talk to each other, genes have multiple inputs, regulatory circuits can be very complicated. I was trying to get a map of the New York subway system, and that's the way it really looks. Everything touching each other. And what we don't understand is how, if so many things are touching each other, how decisions ever get made. I mean, that's something that's just emerged in the past few years. How with so many multiple inputs and genes talking to each other, how cells actually finally decide on a given pathway. So the main point is that cells become different from each other, these muscle cell and the brain cell coming from a fertilized egg. Of course, because they have different RNA molecules, but they have different RNA molecules because they inherit from their mother different regulatory protein molecules. Now, what I want to do is talk a little bit about a gene that we have concentrated on called MyoD. MyoD is a master gene which seems to turn on muscle development. Now, one of the ways we study MyoD, and a common way is in tissue culture, where we take what's called myoblasts. These are cells that you can get out of your muscle. They're there, and you just grow them in tissue culture. We can grow them stably for many, many years. And when you induce them, they differentiate into muscle cells right there in the tissue culture dish before your eyes. Very important, we can look for cells which have acquired mutations, that is, abnormalities in their DNA sequence for important genes. And those cells are muscle mutants. They no longer differentiate. And what we do is look in the tissue culture dish and say, aha, there's a cell, whereas 1 million of these cells will differentiate into a muscle cell which looks like that. This guy will not. Maybe there's something wrong with an important gene in that cell which makes it unable to do that. And we pluck out that cell, put it in a separate dish, and grow it up and study it. So we want to know what genes are expressed in muscle cells, but not in skin cells or fibroblasts. And most importantly, are there any genes which are expressed in muscle cells, but not working in these mutants of muscle cells? The ones that were muscle cells but have acquired a mutation so they no longer can make muscle. I don't think I'll... And what we did then was to compare the RNA. That is, we want to know what genes are active. The best way to do that is to look at the RNA of a muscle cell versus a skin cell. And we can use tricks to say, here's an RNA, it's in both, get rid of it. Here's an RNA only in muscle cells, let's keep it and test it. And we compare them, one to the other. And you can imagine we can use some sort of hybridization technique to do that. And that's what we did. And then we get a certain set of genes, about 5,000 genes, which are working in muscle cells, but not working in skin cells. How do we know which gene might be an important regulatory gene? The way we did that was to look at these muscle mutants. We made the hypothesis that an important muscle regulatory gene would not be expressed in these mutants. And we found a gene called MyoD. And MyoD has this amazing capacity, as you see here, that if we take the gene and just throw it into a dish, take the DNA, as you saw on that electron micrograph, the pure DNA, put it into the tissue culture medium, a few cells will take up the DNA and express it. Those cells, when we look, turn into muscle cells. That's the red. Now what you see here are a whole bunch of cells that didn't take up DNA. Their nuclei are stained in blue. And then we stain this dish with an antibody to a molecule which is present in muscle cells called myosin. And here you see the red, because the antibody had a red dye on it. So this cell has turned into a muscle cell. And the muscle cell, of course, is characterized by a very characteristic set of proteins which define it, roughly about 1,000 proteins. Among the proteins are these proteins that cause contraction. And they cause contraction by going like this. They slide on each other, as you see here. So you start off with them like that, and then they come together, and the muscle contracts. So these are very specific markers for a muscle cell. And that's one of those markers I just showed you in the stained picture. And this is an electron micrograph of a skin cell which was turned into a muscle cell. And you see these beautiful arranged crosslinks, which when you add ATP contract and cause your muscle to shorten. So it's a very complicated, in one of these units, this is all in one cell. In one of these units, there are about 50 different muscle-specific proteins expressed only in muscle. All of those are turned on when we put MyoD into these cells. So what MyoD does, here's the MyoD gene. It makes an RNA, it makes a MyoD protein. I won't tell you this, but the MyoD protein, as I indicated before, binds to its own gene. It binds to a specific sequence, and that sequence is this, CAGCTG. So it looks all over the genome, all over the DNA. Is that sequence there? It scans. If it's there, it binds. Once it binds, of course, this non-specific protein, RNA polymerase, comes in, copies the gene into an RNA molecule, and now the gene is active. Without MyoD, it's inactive. And that's its state in a brain cell, in a blood cell, in a skin cell. But MyoD does other things. It will bind to all of the genes which are responsible for making a muscle cell a muscle cell. These genes are those genes that I showed you in the products of those genes are those responsible for causing those cross striations. Okay? So it will activate about 1,000 genes. Yeah, sorry. Huh. Well, MyoD is an ancient gene. It's present in sea urchins, in worms, and it has historical precedence as far back as yeast in its basic structure. So it has evolved from other genes, which will also bind to DNA, but to a different sequence. But wherever you see muscle in biology, you see MyoD. Oh, you're talking not in evolution, but in development. I'm going to talk about that. That is the major question. If you want to make a muscle cell over here, where do you get the information to do that? And that's the major breakthrough, I think, in biology in the past 5 years, because we understand how that happens. Now, MyoD has the capacity to turn almost every cell type it is expressed in into muscle. So we can put MyoD into brain cells and turn brain cells into muscle. And this is very important over here. We can put MyoD into fat cells and turn fat into muscle. Yeah. Right. We missed out on that one. I might mention that one of the things that a lot of clinicians have approached me on is putting MyoD into skin cells, okay, and then causing the skin cells to become muscle cells and injecting those muscle cells back into muscle from people who have muscular dystrophy and causing the muscle to regrow without any kind of immune suppression because it's their own cells. Another application that people have talked to me about is actually injecting the MyoD gene, just as we do in tissue culture, into the heart to increase the contractile strength of the heart for people who have heart disease. Yeah? The skin cell, that's a good question. We're not really sure. In most cases, certainly the fat cell turns off its fat program. And pigment cells turn off the pigment program. In the case of in the case of the skin cell, we're not really sure. Yeah.
You showed a 6-base sequence that the MyoD recognized.
Uh-oh, this is going to be a tough one.
By quick calculations, I see that 6 bases would code for about 16,000 possibilities.
Absolutely.
And from your bookkeeping, there are 5,000 different sequences in particular cells, and 20,000 overall. 16 is just between the two.
Yeah.
That means that only the specific ones are bound, and in a very conservative way, and only the specific ones are identified, and the others are not.
Well, it's actually just a multiplexing problem answer. What most genes do is have several signals, okay? 2 sequences. So you bind 2 molecules of MyoD to the DNA, and MyoD has this property that if it's next to another molecule of MyoD, they like that, and they touch each other like this, and they hold each other on, so that if this eventually goes, this one stays, and so on. So it's going like this all the time. So by having 2, you increase the specificity that way. So this is a fat cell which is turned into a muscle cell.
How do you do that?
How? Oh, we took fat cells in tissue culture and added MyoD, and We used the virus in that case, yeah. We took a MyoD, put it into a virus. The virus would infect the fat cells and express the viral genes, among which the virus thinks MyoD is one because we trick it, and it turns into, but we could do it just by editing the DNA. And here you can see a bone cell, which has been turned into a muscle cell, and you can see that with this antibody staining for myosin, which is the muscle-specific gene, you can actually see these cross striations. And if you add an inducer to the medium, you can see the muscle actually contracting in the tissue culture dish. I think I should stop there. So this gets back to the question that was just asked. So we've simplified the understanding. We think we can say, well, in order to make it a muscle cell different from a brain cell, you activate MyoD in a muscle cell, and we know a gene, at least in fruit flies, which is important, which is just like MyoD but slightly different, and it will activate nerve in nerve cells. Thanks. Does it come out again? Oh, well. I'll just... Okay, now the really major breakthrough in trying to understand, so we activate muscle here and over here. Okay, we make a muscle cell appear over here. And we think we know now that all you got to do is turn on MyoD, and when you do that, everything else follows. MyoD binds to these other genes and turns their transcription on, and you become a muscle cell. That's obviously oversimplified. Many problems, as Michael discussed, but that's probably a general solution. The question is, how do we know to put a muscle activate MyoD here, and hopefully not there? Okay. The answer to that is that during the development of the embryo sets up, the early dividing embryo sets up a coordinate system. That is just like the globe, latitude and longitude. So every position is known in the embryo. It's a coordinate system of these regulatory protein molecules which can bind to DNA, for the most part. So if you think of the globe, Italy is, I don't know, gut, obviously. Russia is muscle. England is brain, and that's where you put these things. You read the coordinate system, and you say, activate MyoD in Russia. Okay? And then the next question becomes, number one, you should be asking, how do we know that? It seems amazing, kind of preposterous. And the next question is, how do you read the coordinate system? What is the coordinate system made of, and how do you read it? And once you read it, you activate a gene like MyoD for muscle in this position. Okay? Now, the best way this problem has been studied is in fruit flies, because you can do a mutant analysis. That is, you look for fruit fly children who are abnormal. Now, after treating the mothers with awful chemicals, that DDT and so on, which cause the DNA to change and cause the gene the gene not to work. Now, among the kinds of mutations that you see when you do this, and look very early in embryogenesis, are flies, let's say, with no heads. or flies with a head here and a head here, or flies with, no abdomen. And that has identified certain genes and allowed the players in the game to be known. The result of that is the following kind of model, which is absolutely correct. That is, the egg does not develop in isolation for the most part. It develops in a womb or a follicle. And it's the asymmetry of the follicle which imposes on the developing egg a coordinate system. So here you see the follicle around the egg, and the cells in the follicle over there actually pump in the RNA for a gene. They pump it in. They make it, the egg doesn't make it, it's the mother. The mother's womb is making this RNA. It pumps it into there, and it sort of sticks there. And it forms a gradient as it diffuses this way. That establishes the anterior coordinate up here. Similarly, over here on the right-hand side, those cells in the womb put in the orange RNA. And that diffuses upward, establishing posterior. So this is latitude. Over here, the cells down in the bottom send a signal. I'll get back to that. To the bottom. Yeah?
Is the RNA being transcribed to the cells within the egg?
No. The RNA comes from the mother. The RNA comes from the mother from the womb, from the cells surrounding the egg. And those cells physically move the RNA into the egg. I'll come back to that. Good question.
What kind of RNA is it?
It's an RNA that codes for a protein. And that protein is a regulatory protein just like MyoD. It binds to DNA. So here the mother puts stuff there. It puts stuff there. It sends one signal on the bottom and one signal on the top. So you establish an axis, anterior, posterior, top, bottom, dorsal, ventral. Okay? And the consequences of that are latitude and longitude, where latitude is defined as because diffusion is occurring, there's a gradient this way, a gradient that way, a gradient that way, and a gradient that way. So every position has a value, and I'll refer to that as positional value. And what you do is read the coordinates by the level of purple, yellow, orange, and green on the top. Yeah. Yep. Yep. 2 different RNAs, 2 different protein products. They bind to 2 different kinds of sequences on the DNA.
Not 4 different RNAs, just 2.
Oh, 4, yeah. I'm really simplifying this just to get the notion of coordinates across. And what you do if you want to activate muscle over here is read the coordinate system, and MyoD gets turned on. Brain over here, read the coordinate system, and a gene called Akit-Scoot, which is a master gene for brain, gets turned on here. And that's how you get cells in the right positions.
Is there a function of distance there? That's two-dimensional, the way in which the coordinates are established, but the egg is three-dimensional, something further out as opposed to closer in to the
Yeah.
axis.
Yeah, I've, of course, left out what's happening inside, right? The gut, the internal muscle. And that, I haven't thought about that much, but what probably happens is that during gastrulation, some of these cells over here will move inside and establish the third coordinate that way. So that's the general idea. Now, I told you before that cells talk to each other. And talking to each other allows them to refine this coordinate system. Now, what do I mean by talking to each other? Again, there are many ways they can do this, but there's a very physical way. One way you all know about. Progesterone is a small little molecule of hormone. It's produced, I guess I don't remember where. It's produced somewhere here, and it eventually circulates through the system and finds itself, for example, in the ovary. And what it does there is to go through the membrane, bind to a protein, and that cause that protein then to enter the nucleus, bind to specific DNA sequences, and turn on certain genes. So that's one form of communication. There's another form of communication where, for example, two cells come together, and this cell tells this cell by direct touching to do something. I'll show you a particular mechanism. This cell will have a so-called receptor on it. That's a protein which looks like this. And at the base of the protein inside the cell is a regulatory protein. Let's say the orange guy. When this cell touches this cell has on its surface something that looks like this. And this is the way these things go. We know the atomic structure of the way these things touch each other. And this guy fits right in here. The two receptors now come together, surrounds this inducer in red. As a result of that, the orange regulatory proteins are released. They go into this nucleus, and for example, they will activate a gene like MyoD. So that's how cells talk to each other. So what you can do during development and in the establishment and further refinement of this coordinate system is you have two cells, A and B. B talks to A. When A divides, as a result of that conversation, it generates a third cell type called C. And now C can talk to B. And when B divides, it generates another cell type called E. And this kind of logic and propagation and refinement can help establish the pattern. And this is an early Drosophila embryo. And what one is doing is detecting specific regulatory proteins with antibodies dyed with different colored dyes on it. And the different stripes you see represent the different proteins and where they're positioned. And you can see that it's a beautiful array of information. You can't tell that these cells are different at this early stage in development, but this is the coordinate system that will eventually help to encode where in space particular cell types should arise. I'm not keeping track of time. Right? Okay, so you don't believe me. So this is an absolutely beautiful experiment that unfortunately I didn't do. What was done here is, you remember I told you that in this part of the molecule, the mother, which you don't see here, has put in RNA from the gene, for a specific gene. This gene is called bicoid. And that determines the anterior pole of the cell, in this longitude latitude model. Well, if you make the mother have one copy of bicoid and look at where bicoid protein is expressed, you see it's all over there. So even though the mother, whose cells surround the whole follicle, the whole egg, has one copy of the bicoid gene, it's only put into the egg at this end. And that's as far as it diffuses. Here, the mother is tricked to having 4 copies of this RNA. The embryo, using different genetic tricks, has only 1 copy. And you can see now that you're making more bicoid protein, and it diffuses further. As a consequence, these stripes that I just showed you appear, because they're reading this gradient, appear further down than over here. So these stripes are responding to this gradient. And if you chose a different gene which is responding, you'll get stripes in the middle here. Boom, 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 boom. And they'll be moved to the right relative to there. The details of how the coordinates are made and established and propagated and maintained are just now being worked out. The important point is that I think everyone would agree on is that there is some sort of coordinate system, and genes like MyoD, which make muscle, read that coordinate system and activate muscle in particular spots. Question? I'll come back to that at the end. Any other questions?
What do you remember what end of the egg cell, the RNA?
I'm glad you told me that. What you can do experimentally is inject in RNA. And if you inject in RNA over here for that gene over there, for bicoid, what you get then is an animal with 2 heads. Okay? If you inject in the RNA for the posterior part, called nanos, then you get 2 tails. If you inject in RNA, which makes the dorsal part over here, if you inject it over here, you get 2 backbones. That RNA back here is called dorsal. And it was originally found because the embryos that came out did not have a backbone. Well, they of course didn't have a backbone, but they didn't have the dorsal tissues. Yeah.
At what point in the development of the ovum is this injection of the RNA?
Oh, you have to do it very early. You have to do it very early. All this is occurring within about 15 minutes.
At what stage? Does it have to be complete ovum, or is it earlier in the meiotic process?
They're usually done in the right after fertilization. That's, yeah. When you inject, because things don't start until after that.
I'm confused. I was under the impression that this was from the mother and it was put into the ovum or the egg before fertilization.
It's put in before, but it actually doesn't start. You don't start until after fertilization. So you establish this gradient before fertilization, and it's stable. And then you fertilize, and then the clock starts. And then you have to respond to it within X amount of time. Now, the truly amazing thing is that when you look at the molecular details of development in different organisms, and even in the fly itself, the actual molecular mechanisms are different. in detail. You set up a coordinate system, but the anterior system in detail is different than the posterior, different than the dorsal, different from the ventral. And it's different in the anterior frogs versus flies. So this is this has brought forth the notion, which I think is a really important one, that during the evolution of development, evolution doesn't work by a very directed mechanism. Jacob has talked quite extensively about this, and the idea is that organisms don't evolve as if an engineer were directing this, but as more like a tinkerer. And whatever works, works. And for some organisms, the Drosophila fruit fly pattern is the thing that they've chosen. Frogs, as a question earlier was pointed out, do things a little differently. The net effect is to set up a coordinate system. So in frogs, for example, what happens is that the mother sets up the anterior-posterior axis of the coordinate system, but the sperm entry point, where the sperm enters the egg, sets up dorsal versus ventral. What happens is the sperm comes in, and there's a rotation that then occurs. And the outside of the egg moves relative to the inside, like that. Boom. And that establishes the asymmetry for dorsal versus ventral, and for the unique frog coordinate system. So it's the position at which the sperm enters. So for that, the question that was asked earlier, this is how, and if you parthenogenetically activate the embryo, it's where you put the pinprick. Now, one of the pieces of evidence that as a result of this rotation, this region of the embryo becomes different from the dorsal region. And we know that's the case because if you take this region of the embryo from one embryo and put it into another embryo over here on the ventral side, what you get out are tadpoles with two dorsal ends. Okay? And this is a picture of one of these guys. Here you see one head, one back, and another back. So we know that there is dorsal information in that region of the embryo. Now, here's that region of the embryo, which is something magical has happened as a result of the sperm coming in on that side. There's this rotation. Something funny has happened there, so that this region over here is very different. We know that's different because if we take it out, a little piece of tissue, and take it out, it has the property of being able to induce muscle, make a tissue it touches turn into muscle. And we can show that by taking the top of this embryo, which normally turns into skin. If you take it off, it'll turn into skin. However, if we mix it, just put it right on top of this region, that tissue will now turn into muscle. So the notion is that the yellow guys are secreting something which is inducing muscle in a tissue which will normally become skin. So this is one experimental approach where we can more accurately define what it is that's turning on MyoD. Now, initially, in a frog embryo, just by random noise, MyoD is turned on everywhere in the embryo. And when we found that, we were really surprised, because if MyoD is turned on everywhere in the frog embryo, we would have expected that muscle appear everywhere. And it didn't. We knew MyoD was there, but there was no muscle. And then we looked with an antibody to see where MyoD was. And to our surprise, it was only in the cytoplasm. It was there everywhere, but it was only in the cytoplasm. Of course, it has to be in the nucleus to bind to DNA. And in fact, it was in the nucleus. This is all unpublished. It was in the nucleus, but it was only in the nucleus where muscle was being induced. So where the coordinate system was working to establish a certain coordinate to make muscle, MyoD moved from the cytoplasm into the nucleus and presumably activated genes to make muscle. And this is what I depict here. This is that region which can induce these double embryos, and it's secreting a product which causes MyoD to enter the nucleus. And this is a low power view of that in the frog embryo. We're staining MyoD with an antibody, and if you look over here versus over here, this is where induction of muscle is occurring. In regions which don't normally make muscle, you see MyoD in the cytoplasm, and you see that white spot there is the nucleus. It's not there. And in regions which are making muscle, you see that MyoD is in the nucleus over there. So we think that what's happening is depicted here, that MyoD is made everywhere initially by a non-specific mechanism, but it can't get into the nucleus. It's in the cytoplasm. When induction occurs, that process of turning on muscle, it goes into the nucleus, and these cells now become muscle. Sorry. So we think that one of the upstream activators of determining where muscle is going to occur is the actual induction process, which occurs in frogs. And that induction process is part of the mechanism which establishes coordinates in the frog system. So I want to, how are we doing for time? What? 4:15. Why don't I come back to worms if I have time and get back to the question that was asked. So we have this coordinate system. How does the coordinate system activate MyoD? How does MyoD read that coordinate system? We don't really know in exact detail how that happens. But the way we think about the problem is illustrated here. Sorry. So we imagine that what's happening during embryogenesis is that you set this coordinate system up. Now, x, y, and z in the coordinate system corresponds to 100 units, let's say, of the anterior factor, 50 units of the posterior factor, 20 units of the dorsal factor, and 5 units of the ventral factor. This is the total amount of those proteins that happen to be in this position at the important time in development. Now, if you have that constellation, what happens then? So the actual value of the coordinate system is determined by the concentration or the amount of each of these 4 elements. Once you have that amount, how do you read it? Is that a fair? Everyone follow? Well, you read it by saying on the linear DNA, there is a one-to-one relationship between the coordinate system, the levels of these 4 proteins, and a DNA sequence. So, XYZ in the coordinate system is equivalent to the sequence ATCG. ABC is equivalent to the sequence AATT. So, in let's say, a muscle cell, where you want to put a muscle cell in XYZ coordinate, you have to have upstream of the MyoD gene the sequence ATCG. MyoD reads this the XYZ proteins read this sequence and bind to this DNA sequence here and activate MyoD transcription. This is wrong. This should be a brain cell over here. It would read AATT. Okay? And now this constellation of coordinate proteins binds here and activates brain at that position. Now, obviously, muscle is in many positions. It's here and here and here and here. So let me just finish. So what you really want to do is have not only ATCG determine MyoD activity, but another sequence, let's say up here, say TTTT, which represents a position in that coordinate, and have muscle there. So there are multiple inputs. Question?
Yeah, as I understand it, the difference between any 2 points on the coordinate system is just concentrations, not different proteins.
Well, I've tried to simplify it. The different concentrations would activate intermediary genes as well. And once you activate the intermediary genes, then you can use single proteins to do the job. This is all, again, right at the cutting edge. And how exactly this occurs in physical terms is unclear. That this is occurring is, I think, incontrovertible because we can change the value of the gradient, as I showed you by putting in 1 copy or 4 copies of bicoid and moving the response this way or that way. So that it occurs is clear. How it occurs exactly on a mechanistic basis is unclear. How you read these different levels of concentrations, I don't think we have a clue really how you do that. But nevertheless, the end product of that is to read a DNA sequence. How do we know that? Well, we put a mutation in this to cause it to be TTCG, and muscle doesn't appear here. Okay? So that's how we know it. And I should say that this has not been done in muscle, it's been done with the equivalent gene for nerve in Drosophila. Okay? Question?
There are inhibitors, do these work as inhibitors as well? Like the ones that would activate nerve with inhibitors?
That's the assumption, but I don't think it's been, I don't think, you don't really know where the inhibition might be occurring to prevent this mutual exclusion so that you don't make a cell which is both brain and muscle at the same time. It could be occurring either at this level of reading the coordinates, or it could be occurring because MyoD binds to this nerve master gene and turns it off. So that's not clear. Are there any other questions? So this, I think, is the first major, there are three major points. One is this idea of a master gene, like MyoD. The next point is the idea of a coordinate system. And then this is a little bit more subtle, how you read the coordinate system. And it's a transformation of a three-dimensional coordinate system onto the linear DNA, and you do that transformation by having a particular DNA sequence reading the X, Y, and Z. Okay? Now, a little bit more complication. You say, well, this is all well and good, but what happens in the adult? What happens after you lose all of these molecules that have been put into the coordinate system by the mother? Eventually they'll degrade, eventually they'll pop off the DNA. Well, the cell is very tricky. And what it does is this. So initially you get the gene started by binding here to ATCG, but after this orange guy disappears in development, what you do is put in a sequence CCGG, let's say, and now MyoD protein itself would bind there and stabilize the circuit, cause this gene to be activated. So there's an initial activation by the coordinate system, then you get rid of that, and then MyoD keeps itself on by this autoregulatory circuit. It binds upstream, keeps itself on. So you no longer need the orange guys to keep the gene going and to make sure muscle always stays here in the right spot. It's an amazingly reasonable system. Okay, the last slide is the really emphasizes the importance of mom. The mother is really the thing that keeps everything going.
We knew that.
Yeah, I was waiting for that. I'm a straight man here. So this is an attempt to try to show you how this is occurring, how there's a cycle. So here's a mother, and here's her womb. And the egg develops here, and here's the womb around the egg. The cells here, these are part of the mother now. These cells in the womb are the mother. They're like muscle or blood, it's just a womb. They're so-called somatic cells. These cells here are different from those cells, are different from those, and different from those, in only a very slightly different way. These guys can make orange and put that into the mother, the egg there. These guys make yellow and put it up there. These guys blue, let's say, and these guys down here purple. So the coordinate system is established. As a result of that, you get an asymmetric egg. The asymmetric egg is shown up there, and father comes along, fertilizes the egg, and the egg, because of its asymmetry, can generate a new mother, as I tried to show you before, how you read this coordinate system. And as part of the information for generating a mother, just like you generate a muscle cell, you generate an asymmetric womb. The developing germ cell, the fertilized egg, gets inserted into the womb, and the cycle continues. So you go round and around. It's a closed circuit. The mother, asymmetric womb, asymmetric egg, mother, so on and so forth. And this, I think, is what I've tried to do is give you a much abbreviated and simplistic kind of logic to how gene regulation occurs, how you generate differences in a fertilized egg by virtue of having a mother generate those differences for you. And I haven't dwelled very much on the specific mechanisms. How exactly, when a protein binds to DNA, does it recognize the sequence CAGGTG? What happens there? What are the actual chemical bonds that occur which cause the protein to bind to that sequence? Once it binds, what happens, how does it recruit RNA polymerase to come in and transcribe, make copies of that gene? These are all questions which are now being answered in excruciating detail. And we know an awful lot about that, and maybe some other speaker will address that. And then finally, a very major question that I haven't really touched on is, how do you integrate? You have multiple signals coming into a given gene. Go, stop, go, stop, go, stop. And those are represented by proteins actually physically bound to the DNA. How do they communicate to come up with an output? The gene either has to work or not work. How do they talk to each other? And that, of course, has something to do with how they touch each other and how they, in turn, touch RNA polymerase to let it go or not go. And that's another whole question about which has to be addressed and which is being understood. And then I think another thing that I alluded to very briefly, which I think is really one of the more fascinating aspects that hasn't been discussed much, is evolution and how while the ability to become a muscle cell seems to be very conserved in evolution, it seems to be dependent on MyoD, the way every embryo sets up its coordinate system is so different that we don't understand why that would be so different. And how that turns on MyoD, as I indicated, seems to be relatively conserved. So I think I'll stop. Thank you. Question?
This is probably new to me, probably the coordinate system. There was a researcher that had the name of Gurdon, something like that, the one who buffed out the
That's right.
nucleus.
The guy that did the experiment I wanted to do. Yeah.
And then replaced it with an already differentiated nucleus.
Yes.
So the whole coordinated system wouldn't apply. recycle into a whole new crop?
Oh, it applies, I think, very nicely because he took the nucleus out, but he left the cytoplasm in.
Oh, so that's where the coordinates.
Yeah, and the coordinate system was there, were potentially there. Of course, in the frog and in C. elegans, the worm, what happens is that in flies, the coordinate system is set up entirely by the mother. In flies and in worms, probably, in frogs and in worms, anterior and posterior is set up by the mother. The potential for dorsal and ventral are set up by mother. But dorsal ventral is actually determined by father, by where the sperm comes in. Okay. Yeah.
Already the computing gradients that are responsible for setting up the coordinate system dependent on the kinetic fashion on gravitational forces.
I don't think so, because I think they've sent worms up and frogs up, and they seem to develop okay. At least some of the embryos do. Yeah.
I may have missed something. What part does I like women being all powerful. What part does the egg play in setting up the organ?
What part does the egg itself play? What you mean actually is, what part does the egg genes, the genes in the nucleus of the egg, play?
It's not only the genes, though, right? Could there be something else in the egg other than the genes?
Well, in terms of, oh yeah, there's a lot of stuff in the egg, but none of it is important. It's the building blocks, not the egg supplies the building blocks, the mother the instructions, as usual, and then the father, I don't know. But the father is, of course, very important because he provides the capacity for evolution. Because by having two copies of a gene, it means that you can mutate this guy, right? And hopefully have it do something wonderful in the future. And still preserve the basic function over here in this copy to maintain life. So that's the reason why we have sex. Yeah, it doesn't play much in setting up the coordinate system. Right?
In your tissue culture slide, whenever myelin is expressed, you seem to get striated muscle cells. Is the implication that that's sort of the default muscle type, and to get other kinds of muscles, you have to modify along the way?
The implication, but not proven, is that there are molecules like MyoD which will be responsible for making heart muscle and the smooth muscle. But those molecules haven't been found. The only case where there is an analogous situation is in nerve, where there is a molecule like MyoD. It's called a Keats scoot, and that's important for making nerve in flies.
If this theory is correct, when the salamander grows a new whatever, what turns on the gene then? Because then you don't have the coordinate system to put it on.
I think, so if you chop off the hand of a salamander, it'll regenerate. And what happens, I think, there, it's controversial again, is that it looks like there are cells there at the stump which are not yet making muscle and bone and cartilage, but which are prepared to do so if given the right instructions. And when they do that, they regenerate a whole limb. And your question is then, how do you get form? How do you get coordinates out of that? Sure, you can get cell type out. And that comes from a refinement. That's not the initial coordinate system. What happens probably is that as these cells, which can differentiate into muscle, bone, and skin, grow out, they talk to each other in the way I illustrated. And one will say to the next, I'm on the right, you're in the middle, and the guy next to you is on the left. And the way they do that is by sending these local signals. And they do that. I mean, I'm not making this up. We know those molecules that are involved in that, and they say, I'm on the right, I'm going to be a muscle. You could be a muscle if you were on the right. That is, this has MyoD, this has MyoD. But since I'm on the right, I'm going to become muscle. I'm going to turn you off. And they send a signal this way and say, hey, you turn yourself off. And then MyoD disappears, and then they go on and make skin. And that's a general outline of how that works. And that's certainly the case over here with hair. You know, if you look at hair development early on, or the equivalent feathers or scales, you get this beautiful packing of a hair in the middle and 6 non-hair cells around it. Hair, 6, this hexagonal packing. And that occurs because the guy in the middle sends a signal to the 6 guys around it and say, you know, you can be a hair, but you better not be. So that's a further refinement on how you, it's a further refinement after you make initial decisions, then you fine-tune it. Yeah.
MyoD in the skin cell and the fat cell. Have you tried it with any other type of cell?
Yeah, pigment cells, and they turn into muscle cells. Well, I think it is inevitable. And something I didn't mention, God, there's all these things I didn't mention, you're not going to believe anything, is that even though you make MyoD and express it, because it is such an important gene, the cell has all sorts of different controls saying, you know, are you sure you want to be a muscle? Are conditions perfectly right? And therefore MyoD is controlled, the protein itself, whether or not it can bind to DNA, by multiple other elements of physiology. You have to have serum or blood around in order for it to work. You have to have the right level of sugar and glucose and so on. So the cell doesn't really decide that it's going to make muscle until everything is just perfect. And it reads that by actually having molecules read the level of ATP in the environment, and if they can bind ATP, they'll bind MyoD, and MyoD will now work. And there are a whole bunch of signals into that aspect. So when you ask, does MyoD work in every cell type? It works in most cell types, but there are certainly some cell types, like, for example, the early frog embryo. MyoD can be put in, doesn't work at all. The reason it doesn't work is that it gets stuck out in the cytoplasm. So there are all these other kinds of controls which feed in on it. Yeah.
Is there a possibility of a brave new world?
If you voted for Clinton, I suppose.
As in Aldous Huxley.
The question was, is there a possibility for a brave new world? So I meant that in a positive way, I'm sorry. I think what you're getting at is there a time when investigators will go in and change genes. And of course, they're doing that now. There's Dusty Miller at the Hutchinson Center has been putting genes into people now for about a year. And the gene is it's called adenosine deaminase. And people who have Lesch-Nyhan syndrome, I think that is, can be cured by this gene. And, I think that's a good idea, if it can be worked and controlled. I think taking genes out is going to be harder. But, certainly for disease, I think it's something that we should be doing. For making people better, I think that is a political question. Yeah. Thanks. Yeah, thanks.